In this presentation, we'll be talking about Varicella zoster virus, an important worldwide viral pathogen that has some unique characteristics. These pictures show two different types of rashes from two different diseases that are caused by the same virus. The Varicella zoster virus causes two distinct diseases, varicella and zoster. But you're likely familiar with their more common names of chickenpox and shingles. Varicella zoster virus, often shortened to VZV, is a human alpha herpes virus that is in the same subfamily as herpes simplex virus 1 and 2 and is a member of the Varicella virus genus. VZV is a highly communicable disease and occurs in all parts of the world. In this presentation, we'll explore the structure of the Varicella zoster virus, the types of human cells it infects, how it replicates within the cell, how the human immune system interacts with the virus, and how one virus can cause two different diseases. First, let's look at the structure of the VZV virus. The VZV virions are about 80 to 200 nanometers in diameter and spherical. In the center is the core. This stores the DNA, or genetic material. VZV's DNA is a single, linear, double-stranded molecule that's about 125,000 base pairs long. Next is the capsid. The capsid is a protein coat that surrounds the DNA. VZV has a 100 nanometer nucleocapsid of 162 hexameric and pentameric capsomeres arranged in an icosahedral form. The capsid is loosely surrounded by proteins called the tegument. The tegument contains the viral enzymes needed for initiating viral reproduction once in a host cell. Finally, there's the envelope, the outermost layer of the virus that is made from modified host cell membrane. VZV's envelope is studded with glycoproteins that are each approximately 8 nanometers long and bind to the specific host receptors and mediate virus entry. Traditionally, the virus was thought to spread to others from the respiratory tract secretions of an infected individual, but there's actually not been much evidence to support that theory. Instead, most virus is believed to come from the skin, where it is highly concentrated in vesicles. Skin cells frequently shed and are probably the primary source of infectious cell-free airborne virus. Interestingly, infected individuals without skin lesions are not contagious to others. Upon entry into the human host, VZV first infects mucosal epithelium of the upper respiratory tract, and it proliferates in regional lymph nodes of the upper respiratory tract, as well as the tonsils. During primary infection, infected Langerhans cells, along with the mucosal and dendritic cells, carry the virus to T cells located in the lymph nodes. Infected T cells express skin homing factors and transport the virus to dermal fibroblasts and keratinocytes, which produce pro-inflammatory cytokines, resulting in the characteristic skin eruptions of pox or varicella. Despite a robust immune response and the eventual disappearance of acute symptoms, VZV is never completely eliminated. The virus remains in neurons in the dorsal root ganglion, where it can lay dormant for decades. During reactivation, the virus travels from the sensory ganglia to the skin nerve terminals, where it infects and replicates. When VZV returns to the skin during reactivation, it goes to the area affected by the specific ganglia in which it lay dormant. This is in contrast to the primary infection of chickenpox, where the rash appears across all areas of the body. The reactivation of VZV illustrates the hallmark feature of the herpes virus family, latency. Latency is the establishment of lifelong infection, and this is how one virus can cause two diseases. So how does the Varicella zoster virus actually infect a cell? Remember in the beginning I mentioned that VZV envelope is studded with glycoproteins. Well, those glycoproteins help the virion attach to the cell. Upon adhesion, receptors on the host cell initiate endocytosis of the virus, allowing it to enter the host cell. Once inside, the virus loses its coating, and the viral core and tegument proteins are released into the cytoplasm. The uncoated capsid is transported into the cell's nucleus, where the viral DNA is released and becomes circular. Next is transcription, or the building of viral proteins. With VZV, transcription occurs in a cascade that leads to 3 distinct classes of mRNAs and the synthesis of viral proteins that are classified as intermediate early, early, and late. And these names are based on their time of expression after virus entry. Immediate early genes are transcribed immediately after infection and contain the major transcriptional regulatory proteins. The early genes encode for proteins and include the enzymes that are required for replication of the viral genome, and the late genes mostly encode structural proteins. After transcription and DNA replication, the particle assembles and exits the cell. The virion acquires its envelope by budding through the inner lamella of the nucleus, and a secondary envelope when it exits the skin. How does the human immune system react with VZV as it enters the body? VZV is sensed by the innate immune system via pattern recognition receptors in dendritic cells that recognize viral pathogen-associated molecular patterns, or sometimes called PAMPs. Cell surface toll-like receptors recognize VZV particles such as glycoproteins, and these responses can trigger inflammatory cytokine secretion or cell death. Primary VZV infection that includes varicella is followed by the production of VZV-specific antibody and VZV-specific T-cell mediated immunity. A key player in the human immune system are natural killer cells. These innate cytotoxic lymphocytes play a significant role in the immune response against most viral infections. Natural killer cells can restrict VZV replication by secreting antiviral molecules such as interferon and granulysin, or by direct cytolysis. The interferon produced by activated natural killer cells promotes clonal expansion of antigen-specific T cells. However, natural killer cells can also become infected and play a role in the distribution of the virus. While the ability of VZV to infect T cells has been well established with previous studies, the ability of VZV to infect natural killer cells has not been scientifically determined. In 2018, a group of scientists set out to understand how VZV directly interacts with natural killer cells and to confirm the belief that VZV is capable of infecting human natural killer cells. To conduct this research, science looked for transmission of infection to human primary peripheral blood mononuclear cells through a co-culture with human epithelial cells that had been infected with an isolate of VZV. A cell-associated model of infection was used to transfer infection to the PBMCs, as VZV is a highly cell-associated virus. By examining expression of the VZV surface glycoprotein complex, glycoprotein E, glycoprotein I, on live lymphocytes by flow cytometry, the study was able to demonstrate an average infection rate of 8% of T cells. This was very consistent with previous studies that had established VZV's ability to infect T cells. But this study also reported an average infection rate of 14% of CD3+, CD56+ lymphocytes and 42% of natural killer cells. This study also explored the preference of VZV to infect specific types of natural killer cells. There are 2 main subsets of natural killer cells differentiated by their expression of CD56. CD56 dim natural killer cells are the most abundant type of natural killer cells in the blood, while the far less abundant CD56 bright cells are found in the tonsils and lymph nodes. It's believed that CD56 bright cells mature into CD56 dim cells, and data from this study also illustrated that VZV consistently favored mature CD56 dim cells for infection. This figure is showing the flow cytometry plots of both mock or control and infected PBMCs. The figure is showing the data for surface VZV glycoprotein E and glycoprotein I expression on live T cells, which you see here, on CD3+, CD56+ lymphocytes, and on natural killer cells. As expected, in the mock or control group, there is no infection registered. No color is found in these boxes. However, infection does occur in the VZV column, in the T cells, in the CD3+, CD56+ lymphocytes, and where you see the greatest expression is among the natural killer cells. And these figures are showing how CD56 dim natural killer cells, remember those are the more mature natural killer cells, exhibit surface VZV glycoprotein E, glycoprotein I expression at a much higher rate than with CD56 bright natural killer cells. This supports the idea that Varicella zoster virus prefers mature CD56 dim natural killer cells as they're more effective at supporting VZV infection. This study began with confirming the theory that VZV infects natural killer cells and added some specificity in terms of which types of cells they prefer. But like any good study that answers questions, it also leaves new questions to be explored. Possible follow-up inquiries would be, does the fact that VZV prefers mature CD56 dim natural killer cells make a difference in the severity of varicella in adults who might have more mature lymphocytes in their system? And also, since natural killer cells roam the body, what role do they have in transporting the virus to the skin and other organs? So in summary, Varicella zoster virus is a worldwide virus that causes chickenpox and shingles. VZV and other members of the alpha herpes virus family are distinguished by their ability to establish a latent infection with the potential to reactivate and spread the virus to other susceptible individuals. This lifelong relationship continually subjects VZV to the human immune system, and as such, VZV has evolved a host of strategies to both evade and manipulate the human immune response.